I've led a team that has discovered something that I've been thinking about for many years. And I've been thinking about an idea of substitutions. And what does it mean to be substitution? And what does it mean to be toxic? So I've led a team that has discovered a microbe that can substitute arsenic for phosphorus in its major biomolecules. But let me step back for a minute. All life that we know of requires carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. And it uses those 6 elements in some of the critical pieces I think we're all familiar with, including DNA and RNA, or the information technology of the cell, the proteins, which are the molecular machines, and the lipids, which separates you from everything else. And so we've discovered an organism that can substitute one element for another in these major biomolecules. So I want to put that in the context of the who, what, where, and how we did this, and give us a little bit about, in an astrobiological context, or life in a planetary context, of what this could mean to us on practical and also a bit more esoteric levels. I'd like to introduce to you today the bacterium GFAJ-1. These are not little potatoes. They are a microbe that scientists lovingly call little bugs, but they're not bugs. They're microbes. And this is a bacterium that, although looks ordinary, and this may look like a type of micrograph many of us may have seen in different places, but it's doing something extraordinary. And so we'll talk about that. But first, let's find out where this microbe is from. Here we're looking at a map of Mono Lake, California. It's in Northern California and east of the Sierras, just outside of Yosemite National Park. It's a very interesting environment, and we're gonna take a look at that. If we could please roll that footage. Mono Lake is 3 times the salt of seawater, a pH of 10. It's basic, like bleach. And it's got very high levels of arsenic. And it's teeming with life. So the seemingly inhospitable environment teems with life, like bacteria and algae and brine shrimp, and is a major stopping point for migratory birds on their way through the United States. And we went to look for an interesting microbe, and we went to an unusual place. So let me tell you a little bit about how we did this. So if you want to look for an organism that can substitute one element for another, you might want to think about where that particular element is abundant. And Mono Lake is abundant in arsenic. But why would I come up with the idea of substituting arsenic for phosphorus? If we think about the periodic table, arsenic lies just below phosphorus on the periodic table. And so they actually have the physical size of arsenic and phosphorus are very similar. We call it the atomic radii. But it's the physical size of the atom, as far as we can tell, is very, very similar. And actually, the fact of this chemical similarity, and there's other things I'd be happy to discuss at length with folks, but that chemical similarity lends insight into something that arsenic is toxic because it looks like phosphorus. So your cells and my cells and microbial cells, they can't tell the difference. And that's very interesting to me as a biochemist. So I went to an environment to look for this particular microbe, and what we did, we took the muds of Mono Lake that we just were introduced to, and we wanted to see if anything would grow. If we took that mud and we gave it in a laboratory environment that was rich in everything else it needed, sugar, vitamins, not that bad for us, and we added no phosphorus and added very high doses of arsenic. It was a double whammy, you could think of it. This is not an experiment that most people might run, but it was driven by my question, is there a microbe on Earth that could substitute arsenic for phosphorus in its basic biomolecular constituents? And so what did we find? We found that not only did this microbe cope or deal with the toxicity, we might say, with arsenic, but it grew and it thrived. And that was amazing. Nothing should have grown. Put your plant in the dark. It doesn't grow. So something grew. Now we wanted to find out what was happening. So we measured the insides of the cells. We took the cells and we measured the total arsenic concentration inside the cells. It was taking up this arsenic. That's unusual. And then we found that the arsenic was associated specifically with a band of genomic DNA. And so we isolated the genomic DNA. I think a lot of us have heard this kind of thing. And we measured that there was arsenic there. And then we could tell that the arsenic wasn't just stuck. It was in an analogous type of chemical environment, or its nearest neighbors looked like it was behaving like phosphorus. So it was associated, it was inside the cell, it was somehow associated with the DNA, and it had this chemical environment, or it had an analogous sort of, it's like sitting at a dinner table and you and your neighbors and how we might see that you were all around. Well, what should be in the place of phosphorus looked like it was arsenic. We measured it as arsenic. And so let's look at an artist's rendition of what we think is going on in the cell. Let's roll that animation, please. So here we're seeing the beautiful, elegant structure of the double helix of DNA. And what I want to highlight is the phosphate backbone, we say, and that's the light orange balls. And it stitches together as we see the edges of DNA. It holds together the DNA, the backbone. And so what we think is happening, what all the evidence we've collected suggests, is that instead of these, we'll see these orange, light orange balls disappear. And represented by green balls, we see that arsenic would be substituting for phosphorus in the backbone of DNA. And you can see how critical this component of the DNA might be. So what I've presented to you today is a microbe doing something different than life as we knew it. I was taught as a biochemist that all life on Earth, all life we know of, to harken back to the pale blue dot ideas of Carl Sagan, all life we know of is here so far. And if there's an organism on Earth doing something different, we've cracked open the door to what's possible for life elsewhere in the universe. And that's profound, and to understand how life is formed and where life is going. This microbe substitutes arsenic for phosphorus in its basic biomolecules. And what else might we find? What else might we want to look for? Thanks, Mary.
Thank you, Felisa. That's very exciting, of course. We have several other people up here and joining us from Arizona. The next person who will be speaking is Dr. James Elzer. He's a professor of ecology, evolution, and environmental sciences in the School of Life Sciences at Arizona State, joining us from Tempe. His background is in ecology, and he's going to put into context the importance of phosphorus and why this substitution of this is of interest. Jim?
Hello, everyone. Greetings from sunny Tempe. It's a very exciting day. Congratulations to Felisa and her team on this really stimulating report that's coming out. And someone who's studied phosphorus for a long time, it's really quite surprising that we're having this discussion. So what I want to do for everyone, though, is try to place phosphorus in the context of ecology and the environment and evolution and how important it is for human beings and